So I'm less important than the others, and I deal with nutrition. Many colleagues know me. I'm part of a foundation, DD Clinic Research, which studies the relation between neurodegenerative diseases of environmental toxicity and what we can do with our diet. And there was a guy who died because of Dabrioni disease. So maybe except for food infections, this is the only disease in which a type of food can cause death. So a part of our group is made of very young colleagues. They are all biologists. The majority of them are biologists. There are 60 people working on this in this institute, so we collect clinical experiences, and we invite university to do some projects. So we go from the territory to the clinical practice. We study positive biology. We study the elderly, but we study the elderly who do not live in protected communities such as Okinawa or Japanese people or other people. We study the elderly who live in contaminated areas. So if I have a person who's more than 100 years of age who lives in Taranto or in another cities which are polluted and so we try to understand why these people have such a long life, and so we try to copy the metabolism, the habits, the survival of these elderly people. And so based on the territory of where they are based, we look for responses, waiting for the institutions to do something. So meanwhile, when mothers come to our office and ask what to give to their children, can we give them the fruit of our fields? So we try to give an answer to all these questions. This beautiful work, which was published in December 2018, in quite an important journal, states what we have been stating for some time. So these people took human bacteria, human microbiota, they transferred it on mice, and they have seen that even if arsenium was injected into these mice had a death rate which was lower, and so a specific pattern of intestinal microorganisms can be copied, and this can protect us, detoxify us from a metal. After listening to our French colleague, I know that metals migrate. So metals move through a magnetic field, and so these bacteria, the microbiome, can change this field. So if there is a metal which is in the bone, I can have no problems even if I've a lot of metal. Mercurium, for example, is neurotoxic. If I have 1 nanogram of mercury in the wrong neuron, while this causes important diseases. So it is important to know where it is and if it moves. Now, the problem concerning this bacteria is that these cannot be sold. They are the so-called anaerobic bacteria. So depending on the gastrointestinal trait. So in our intestine we have three big things. We have a number of neurons, which is equal to the brain of a dog. The main neurotransmitters stay there. We have 4/5 of the residential immunity. So when we talk about immune systems, we talk about blood and lymph nodes, but the majority is in our intestine. And the third element is the organism, which weigh 1 kilo. So there are a lot of parasites and bacteria which in our body, and so they are anaerobic bacteria. That's why we give a lot of importance to raw food. So the best probiotic, which is the best for us, can protect us. So the most important bacteria are fecal bacteria, the bacteria that sometimes we try to destroy through antibiotic therapy, because we um, because there are problems. Constipated, but actually there are some strains that have to be modulated. So the concept of heavy metals that can be taken through a drug, food, is just a biomarker of damage. And if I can trigger those mechanisms which have me put it out of my body, then my defenses can excrete all the rest. When everything we throw away passes through the traditional mechanic barrier, the immunologic barrier, and so the immune system of mucosa and submucosa, and especially the epithelial barrier. So these junctions that you all studied, which extend and retract, are studied a lot today, because you have all heard of the intestinal hypermobility. We don't know what it is. We have no test to measure it. We don't know what causes it. We don't know how to treat it. What do we know? We see that whatever comes to the mucosa finds a place to an access among the mucosa. So it goes down, it triggers a complex immunologic response, and so the cell-mediated immunity, up to a constant subcontinuous activation, which is the infection of the submucosa. Why do oncologists, why are oncologists interested in this? Why is the metastatic cancer never operated? Because it can go all over the body, because we have a lot of lymph nodes, and so the submucosa of the gut of this part of our body is the place in which our immunity, our immunologic tolerance, and so that which has to be tolerated and that which must not be tolerated, once the imprinting is already given, develops here. And so whatever I put away has an influence on the bacteria, and influences if my immune system doesn't recognize a cell which transforms it or not. So nutrition is important. We have evidence in literature. This slide shows all the cytokines which are involved, and showing you these two papers on agroeconomy journals, which show us what immunologic distraction is, what this concept is. You know the difference between the different type of wheat, but in practice, the wheat we have today, why is it different from the wheat they used to eat 70 years ago? I didn't know that, for example, there were differences. Those who selected wheat in the past, the farmer. There was a selection done by the farmers. Today, who selects wheat? How can I decide whether a wheat is better than another one if it has a higher amount of carbohydrates or proteins. So this selection, which the farmer used to do in the past, was done by biologists, geneticists who crossed the different species of wheat. So today we have had several grafts which transformed the genotype, and so agronomists said this. This is a paper that shows the differences in gluten protein composition between all the modern durum wheat genotypes in relation to 20th century breeding in Italy. So high molecular weight alleles were included, the gluten in gliadin index has changed, and the old set of gliadins disappeared. So the phenotype of wheat has changed. And what have they done? They took the polysaccharides taken from this wheat, and they tried the wheat bran, and they tried to give it to mice. So they took the mice, they gave them this food, and so a food was found which in immunodepressed people stimulate immune system. So this type of wheat bran increases the prostaglandin 2, the PGE2, which are the ones of the inflammatory way, and so they change everything. So mice are stimulated and they are no longer immunodepressed. When I introduce 8, 9 or 10 micro portions of this food in my diet, I have a continuous stimulation of these cytokines, which are in submucosa, because it is in the submucosa that they pass. Do you remember how DNA is cut? Well, it is cut where the polymerases recognizes some bonds. So if I change a bond, the polymerases change. My digestive enzymes cut, not by hazard. So if I change the sequences, the cut sites, the fragments are split in other portions, which probably are smaller or are different. They pass where they must not pass, and we don't know what happens. So this is an example of immunologic destruction of submucosa. Once barriers are enlarged, and maybe this is the mechanism of the syndrome of of the intestine. So if there is a stimulus, it is absorbed. So these substances do not stay well, we find them in the cells, and so if they stay, there can be a damage. So controlling this mechanism is fundamental, because I can control this mechanism. First of all, I can reduce these elements. I can reduce these junctions. So we try to study food. What is our wheat? What is our oil? What are our vegetables? But wheat must be Italian. I mean, it must be Italian, and it must come from southern Italy. It must grow in a climate which is not humid. If I import a Canadian wheat or North American wheat, rice coming from Russia, where it is stored in silos, it comes to Italy full of fungi, microtoxins, such as a very famous one, which is the DON, which eats the clothing and occludins which extend these parts which are already enlarged. And so wheat has to be autochthonous, but wheat has to be Italian, has to be cultivated in Italy. Without wheat metals, because we deal with Terra dei Fuochi, the land of fires, where there is the problem of dioxin, but we cannot trace all the substances which can be buried in the field. We need a biomarker, one or two biomarkers, and toxic minerals, mercury, cadmium, and others. Well, they have been easy to dose and to monitor over time. So what did we do? We started with 20 families, so a very small number, statistically not statistically significant. Well, they did a study with 2 rabbits, so maybe this is a good study. So we gave a special diet to these people, so a system which provides for the modification of some bacteria and the activation of autophagy, and so the cellular reparation. And in these people who live in high-risk areas. The first thing we did when we recruited, enrolled these subjects were volunteers. Because we work at the university, we self-finance ourselves. We perform several pilot projects, and these people were scared of eating fruit and vegetables. They were very happy, they felt safe when they went to the supermarket and bought only snacks, or to go to the McDonald's, because there was a clear traceability. I can even eat junk food, but I'm sure that it was not prepared on radioactive barrels. So we were not able to convince people. In the questionnaires, you see the basic diet of these people is based on bread, cheese, and white meat, but there was no fruit and no vegetables. If they were not sure that fruit came from the big supermarkets from Switzerland or other countries, they didn't eat fruit and vegetables. So why did we give the intermittent fasting to these people? Our French colleagues spoke about autophagy. The use of this of juices, for example. Why do we use these juices? Because it is pressed vegetable cell. So it is a mix which is as close as possible to the human stem cell. It's rich in vegetable hormones. All the hormones we studied. And they are very useful things, because when I give hormone to the plant grows. If I give another type of hormone to man, well, humans don't grow. So I cannot have humans eat 10 kilos of fruit and vegetables. And so juices were a way to add a type of substance that I can give during a fasting, in which I give a vegetable cell which is rich in bacteria and which have to change a food that can have problems. And so these are useful to stimulate bacteria that I cannot stimulate through a pill, or the patient who performs a CAT scan a month or every 3 months, or oncologic patients who have chemotherapy. So when you need chemotherapy for a few days, then you no longer need some metals, and so this protocol is applied when drugs are not used. So our foundation is extending. It is taking part in European projects to get funding. and to get to a statistically significant number, just to say that if I live in Taranto, if I live in Bari, if I live in Bologna, or in the land of fires, there are specific types of contaminants. I'm forced to eat some food rather than another type of food, and I have to demonstrate that diet works. It would be better to study these detox mechanisms because they have no cost. I can use food therapy strategy, and so I can change the immune system. I'm referring to pregnancy. I'm referring to important drugs and also to vaccines. Thank you for your attention.